Hello student, welcome to EPZ Pathshala. I am Dr. Kirti Jain from Delhi Pharmaceutical Sciences and Research University. Today I am going to talk my module, that is concept of targeting and its role in nanotechnology under the paper Nanobiotechnology. So students, let us see what we are going to learn in this module. Drug targeting, components of drug targeting systems, label and approaches of drug targeting, and the targeting systems. What is drug targeting? What happens when we administer the drug with the conventional approaches that leads to the non-selective drug distribution and peripheral drug loss. This results into subtherapeutic dose at desired site. So what is happening with the conventional administration? When you are administering the conventional strategies, the drug is being distributed into the whole body, and there is also loss of peripheral drug. Peripheral drug loss means we want to deliver it to, or we want the higher concentration of drug at the target site where the disease is the disease. So what happened? It is going into the other sites also. So this results into the subtherapeutic dose of the drug at the desired site. Consequently, drug resistance may also occur in this case. So conventional drug delivery systems induces simple, fast-acting responses via oral or parenteral routes. Problems associated with this approach include the reduced patient's compliance, then reduced potencies because of partial degradation, toxic level of administration, increased cost associated with the excess dosing, compliance issue due to administration pain. Hence, to get rid of all these drawbacks, explorations are going on throughout the globe to deliver the drug effectively to the diseased site. This involved development of drug carriers like liposomes, ethosomes, nanoparticles, nanocomposites, CNTs, dendrimers, etc., which are smart enough to deliver the drug in intended concentration or the rate at the intended site of action, and this strategy is known as targeted drug delivery. So what are the objectives of targeting? The basic objective of targeting is to achieve a desired pharmacological response at a selected site without undesirable interactions at other sites, which is the most desirable but elusive goal in drug delivery, and to potentially increase the efficacy and reducing the toxicity of new as well as the pre-existing drugs. The first report on the drug targeting was given by the Paul Ehrlich in his theory of magic bullet, which is an approach to treat disease. In this, Ehrlich proposed that if a compound could be made that selectively targeted a disease-causing organism, then a toxin for that organism could be delivered along with the agent of selectivity. Hence, a magic bullet would be created that killed only the organism targeted. So in this way, what will happen? As the organism is going into a particular site, the drug or the therapy will also go into the particular site, and this concept of magic bullet was to some extent realized by the invention of monoclonal antibodies, because the monoclonal antibodies have the affinity only for a single antigen, and hence they provide a very specific binding affinity. In these two images, you can say on the left hand side there is a targeted delivery, in this that the carrier is being conjugated with the or is linked with the targeting agent. So as you can see in the right side, that to target a specific part you can use such type of targeted delivery system. So the magic bullet concept given by the scientist Paul Ehrlich, whose pic you can see in this slide. Now we'll discuss about the different components of drug targeting. Drug targeting, you can see here, since the early 1960s, many scientists have dedicated their research to the development of different targeting strategies for targeting the drug for improved treatment of diseases. Generally, we can say the aim of targeted therapies is to increase the efficacy with reduction in the toxicity. So you can also call it as a concept of clever drug targeting system, and that includes coordinating behavior of three components that includes the targeting moiety. Targeting moiety does the function of recognition and binding to the target. Then the second component is carrier, which carries the drug, and the third component is the active ingredient or the therapeutic drug molecule, which will give you the therapeutic effect. So a targeting system would be briefly defined as a nanocarrier that carries the attached or the loaded drug to the site of action, with its path being monitored by the attached targeting moiety, which can recognize and bind a particular target. As you can see in the left hand side figure, there is a picture showing nanoparticle. Targeting molecule is being attached to the nanoparticle as well as drug. So the targeting molecule will direct the drug to the targeted site. For example, if we want to treat the cancer, as you may know, there is a cancer of a different sites. For example, if we wish to treat lung cancer, and if you are using anti-cancer drug, the most important problem with the anti-cancer drug is their severe toxicity, and this is due to their interaction with the sites which are non-targeted, or which does not have any disease, means the normal cells which are not cancerous. Cancer is only in lung, but if you are giving the drug that is being distributed in the whole body, definitely if it is being interacted with the normal cell, so the action of the anti-cancer drug is to kill the cell, so it will harm the normal cell. That could lead to toxic consequences. Now we will consider the targeted delivery. In the targeted delivery, you have attached some targeting ligand that has specificity as well as affinity to bind with the cells, particularly having the cancer, as we are discussing in this example is lung cell. So this targeting moiety will deliver this carrier system having the drug to this particular site only, and in this way you can save the normal cells and you can target selectively disease cell, and that is the particular advantage of drug targeting. On the right hand side figure, you can see there is a drug targeting system. The first one, drug targeting system. The second, you can see carrier. In the carrier, that can carry the drug. Inside you can see the second drug is within the carrier, and the last there is a targeting ligand. The technical advantages of targeting. The first, the high efficacy, as we have seen or discussed in the previous slide, that we are selectively targeting drug to the disease site, so that will increase the efficacy as well as this will reduce the toxicity and could lead to, definitely it will be then more cost effective. What happen many times when we want to deliver the drug to this particular site, for example, if you wish to deliver the drug for the brain only, for some brain related disorder, so to deliver the drug in brain is a difficult task, and you will have to give the higher amount of the drug than the required to get into the brain, but that drug will go into the whole body, so you will give the more than the required amount. But if you are targeting it selectively to brain, then you can reduce the amount of that drug. So in this way, this system could lead to be a cost effective medicine. Then shorter treatment times, then you can also get the important advantage of delivering drug to the right place in a body at the right time, in the right dose and for the right time period, and not to the other places where drug could show its side effects. In this slide, we will learn about the levels of targeting. Levels of targeting has been defined on the basis which part you are targeting with the system. So that includes the first order. The first order targeting includes the distribution of the targeted system into a particular target site, that is a organ or the tissue, and this order is known as the organ compartmentalization. For example, liver targeting, brain targeting and cancer tissue targeting. And the second order targeting includes the selective delivery of the targeting system or the drug to the specific cell type, and this is known as second order targeting as well as cellular targeting. For example, targeting of drug to specifically to cancer cells or alveolar cells. The third order targeting includes the specific delivery of the drug to the intracellular site of the target cells, and this is also known as intracellular drug targeting. For example, if you are delivering a drug or targeting it to a mitochondria, lysosome or nucleus. Now, approaches of drug targeting, that includes passive targeting, inverse targeting, active, dual, double and combination targeting. We'll discuss these in detail in the next slides. The first approach in the targeting is passive targeting, and it is achieved by incorporating therapeutic agent into a macromolecular or a nanoparticulate carrier that passively reaches the target organ, and this happens due to its size, that is in the colloidal range, or and the drug loaded within the nanoparticles or conjugated to macromolecules could target cancer cells passively by enhanced permeation and retention effect. Drugs encapsulated in nanoparticles or drug coupled to macromolecules can passively target tumors through this APR effects. The accumulation of drug or drug carrier system at a particular site due to physicochemical or pharmacological factors, or by making use of pathophysiological and anatomical opportunities, is known as passive targeting. There are several pathophysiological and anatomical factors which are involved in this targeting, particularly passive targeting, are listed here. This includes some pathophysiological factors like inflammation or infection, or the enhanced permeation retention effect, that is EPR effect we discussed in the previous slide. Then physicochemical factors, size, molecular weight, then anatomical opportunities, that includes the catheterization, direct injection and chemical approaches including prodrugs and chemical delivery system. In this EPR effect, it means then in tumor vasculature what happened, there is sometimes a leaky vasculature. What happened? Tumor need nutrients more than the normal cells. So that's why there is leaky vasculature that allows the enhanced permeability for the nutrients and the other material also. So that's why it starts with the enhanced permeation, and also the tumor doesn't want to leak these nutrients easily, so they have increased retention effect. So and this increased retention effect is achieved by the poor lymphatic drainage, and this is known as enhanced permeation effect, and due to this effect the macromolecular carriers get entrapped into the tumor vasculature. Another strategy for the targeting is active targeting, and it includes the targeting of the drug carrier using some directing ligand. So basically the targeting is mainly achieved by two types of strategies, that is passive targeting and active targeting. In the passive targeting, the drug carrier complex is often delivered to macrophages and other cells of the monocyte phagocytic system, and this leads to gradual degradation of the carrier with the slow release of the drug from the cells either into the blood circulation or into the tissue environment, or I should say here that liberated from the carrier slowly into the blood circulation or the nearby tissue environment. So by size exclusion, extravasation of carrier drug complex can be limited, and thereby preventing the drug from being distributed to non-target sites. As a consequence, toxicity can be reduced. This is the passive targeting. Now active targeting, that is defined as that active targeting should lead to a higher therapeutic concentration of the drug at the site of action, and which could be accomplished by cell-specific delivery of the drug. In the ideal case, the dose of drug can be reduced and side effects can be diminished. The majority, if not all, active drug targeting strategies exploit receptor-based drug targeting principles, in which receptor-specific ligands attached to the carrier drug complex or directly to the drug itself deliver the drug to the target cell of choice. So depending on the subsequent routing of the receptor complex, the drug will arrive in a specific compartment in the target cell. And specific modification of drug or drug carrier systems with active agents having selective affinity for recognizing and interacting with a specific cell, tissue or organ in the body constitutes the active targeted drug delivery. For the receptor mediated active targeting, you need to use different types of receptors. Here I have given the name of some of the receptors which has been used for the targeting, that includes folic acid receptor, low density lipoprotein receptors, peptide receptors, lipid components of the cell membrane, surface antigens and proteins. The moieties that could be recognized by these receptors includes folic acid receptors, you can see folic acid, then sugars for some other receptors, then you have antibodies, lectins, modified albumins, peptides, etc. In this you can say what is the basic difference between the passive targeting and the active targeting. In the lower part is showing the passive targeting. Passive targeting is due to the special physiology of the deceased part, and this will lead to the uh, attraction of the drug carrier to that disease part. But in the active targeting, you will attach some of the targeting ligand that will direct the drug delivery system to the target site. There you can see there is entrance of nanoparticles, and due to passive targeting, they are gaining into the disease part. In the upper part, you can say due to the attached ligand, that the enlargement showing on the right hand side on the top, you can see there are some ligand that are being bound with that cell, and this is causing the entrapment of this carrier and the release of the drug within the cells. Inverse targeting approaches that includes development of the material that attempts to circumvent and avoid passive uptake of colloidal carriers by the reticuloendothelial system by reversing the biodistribution trend of the carrier. The dual targeting includes the carrier molecules which have their own intrinsic activity, for example like antiviral effect, and hence they will synergize with the antiviral effect of the loaded active drug. In the double targeting, you can say there will be combined spatial and temporal component. What is the spatial and temporal component? Temporal means when you are controlling the time of release, and spatial means that is you are controlling the site where the drug will release. You can get the sustained release as well as the targeted release. Combination targeting includes the carriers, polymers and homing devices of molecular specificity that could provide a direct approach to target site. Some emerging trends in the targeting includes vascular targeting, intracellular targeting, promoter transcriptional targeting, physical targeting, pre-targeting or chemical strategy. In the vascular targeting, you can see it is important because of distinct biochemical differences and easy accessibility by all drugs absorbed or injected in the bloodstream. We mean here by distinct biochemical differences is with the vasculature. Vasculature has some specific structure as well as presence of some specific molecules. For example, like you can target the tumor vasculature via cell adhesion receptor and integrins or some antigens. Intracellular targeting is when you are delivering the drug within the cells. What happens many times the drug, when although they may be going inside the cell, but they could get efflux out of the cell via like multi-drug resistant proteins or P-glycoprotein, which are the efflux transporter, and that could lead to the resistance to many drugs like glucocorticoids, anti-cancer drug, etc. So in that case, you need the intracellular targeting and the silencing of these efflux transporter. Then promotional or transcriptional targeting, that includes the targeted gene therapy, which involves use of a specific tissue, tumor or induced promoter that can limit gene expression to target cells, which express a specific transcription factor. And with this targeting approach, you can restrict the transgene expression in the target tissue, and you can allow the flexibility to regulate the duration level of expression exogenously. Now, physical targeting. Physical targeting includes the targeting via physical means like ultrasound or magnetic field. Okay, in the ultrasound, we use ultrasound to trigger the release of the drug at the target site and to enhance the cellular uptake of the drug. In this, we have proposed the mechanisms like you can use ultrasonic triggered extravasation of polymeric micelles, or the drug release that has been activated by the ultrasonic waves, and you can enhance the uptake into the cells by use of these ultrasonic waves. The magnetic field has been used to localize the gradient to attract the particles to the chosen site and to hold them there until the therapy is complete. So in this, we use magnetic field externally to localize the nanocarrier having some magnetic component, for example magnetite or hematite consisting of iron oxide. So this magnetic field that has been applied externally, that will keep this nanocarrier to that part space for the specific time, and in this way you can attain the effective targeting that could lead to some advantageous therapeutic effect. Next, pre-targeting or sandwich targeting means that we are targeting specific cells with a ligand followed by treatment with a drug linked to another molecule having high affinity for the ligand used. This could include like avidin biotin and streptavidin biotin affinity. And for this, you can include the examples like angiogenesis in tumors, targeted immunotherapy for treatment of cancer. Apart from physical strategies, there are also some chemical strategies. In chemical strategies, we are using some chemical material to target the drug to the disease site. That includes like antibody directed enzyme product therapy. In this, we use antibodies to target the nanocarrier to the disease site. Gene directed enzyme product therapy, that is gene. In this, we will use gene, that includes like gene therapy, and chemical delivery system that is based on the some prodrug mechanism. And in this chemical design of system wherein the active drug is converted into a biologically inert form by attaching targeter and a modifier moiety to it via bioremovable links. That means we are making an active drug into a prodrug that will go inside the body and will be converted into the active form via like enzymatic transformation or some activity of the proteins present on that specific site. Drug targeting systems. In the next slides, we will discuss about the different systems that includes some soluble carriers like monoclonal antibodies. MAVs means monoclonal antibodies, dextrans, soluble synthetic polymers, vesicular or particulate carrier like liposomes, nanoparticles, target specific recognition moieties like again monoclonal antibodies, carbohydrates, lectins, etc. Liposomes, you can say it is like first nanocarrier that has been investigated for the drug delivery. Liposomes could be defined as the small vesicles that are made up of unilamellar or the multilamellar concentric phospholipid bilayers. That could be easy to explain you when we will see the figure of the liposome in the next slide. So with the liposomes are basically composed of phospholipids, and with use of different phospholipids you can vary the charge, lipid composition and the size of the liposomes. Then we have also developed the pegylated liposomes. In pegylated liposomes, what we do, we pegylated the phospholipids, and in this way we can prevent the recognition of liposomes by the phagocytic cells, and that leads to the development of stealth liposomes that have longer circulation time, increased distribution to the peripheral tissues. And further, you can also attach the targeting device or the homing ligand to the surface of liposomes. The areas in which liposomes has been investigated previously, that includes macrophage specific delivery of drugs, passive drug targeting, and non-viral vector mediated gene therapy. In this slide, you can see there has been the different types of liposomes shown in the same figure. The first A, that is top left hand side, you can see conventional liposome. In the conventional liposomes, there are some components that has been shown in this figure. There will be one hydrophobic drug that could be embedded into these lipid bilayers. You can see there are two layers of the spheres. These are showing the phospholipids. Phospholipids basically have the polar head group as well as the hydrophobic tail groups, and liposomes mostly have bilayer of these phospholipids. So in these bilayers have hydrophobic properties, and in the center you can say there is an enclosed aqueous environment. So you can entrap both types of drugs in the liposomes, the hydrophobic drug into the lipid bilayer which have hydrophobic nature or the lipophilic nature, and you can entrap the hydrophilic drugs which have hydrophilic properties into the center of liposome. Further, you can also use positively charged lipid or negatively charged lipid, that is in conventional liposome. The lower side, on the left side, you can see the pygylated liposomes. In this, we pygylated these phospholipid molecules to develop pygylated liposomes, which are also known as stealth liposomes, as we discussed in the previous slide. On the lower side of the right hand corner, you can see there is ligand targeted liposomes. In the ligand, you can say there are different moieties which could be conjugated with the phospholipids. That includes protein, antibody, peptide, carbohydrates, small molecules as well as PEG. On the upper side of the right hand side, you can see there is theranostic liposomes which has been conjugated with the targeting ligand as well as with the imaging ligand. So you can use these liposomes for the diagnostic applications. So the another system that could be used for targeting is monoclonal antibodies and fragments developed by the Kohler and Miles team. And monoclonal antibodies are used in targeting as targeted effector molecules, carriers for selected delivery of drugs, toxins, and also for antigen recognition by antibodies has been explored in the cancer therapy. Some examples of MAB used in the therapy include anti-tumor necrosis factor and alpha, antibodies for treatment of chronic inflammatory diseases, and anti-VEGF antibodies to inhibit the angiogenesis. The lipoproteins, which are the endogenous lipid particles like low density lipoproteins and the high density lipoproteins known as LDL and HDL respectively, containing a lipid and apoprotein moiety, has been used as a targeting agent with the liposomes and some other carriers. And the size and charge of particles determines their in vivo behavior, and hence they have been explored in the targeting of like hepatocyte targeting, and oxidized and acetylated LDL have been explored for Kupffer cell and sinusoidal endothelial cells targeting. Now polymeric micelles, in this you can see they are basically the di-block structure with a hydrophilic cell and a hydrophobic core in the size range of 10 to 100 nanometer. Drugs can be incorporated by either by chemical conjugation or physical entrapment. They have hydrophobic core that is consist of biodegradable or poorly biodegradable polymers, and they also serve as reservoir for the insoluble drug. Further, the surface could be conjugated with the targeting moiety. For example, you can say the polymeric micelles have been explored in passive targeting of anti-cancer drugs to tumors. In this figure, you can say how these polymeric micelles are formed and how they are used for the delivery. So here you can say there is a polymer having the hydrophilic block as well as hydrophobic block. Please see on the left hand side there is a polymer chain shown by the two different colors. One is blue, one is red. The blue part is showing the hydrophilic block and the red part is showing the hydrophobic block. And so we use some strategies then to make the self assembly of these polymer molecules into the micelles type of structure. We have discussed there have been so many advantages of these nanocarrier system, but it is not that much easy to develop such type of material. There have been some of the problems that has been faced or could be anticipated in the development of clinically successful targeted delivery. That includes like immunogenicity, then counterproductive liver clearance. Production yield is very important because many times you use so costly material but you get the very small yield finally. And whether the target cell is of non-hepatic origin, extensive uptake by the liver is not desirable. Then validation of each drug targeting system undergoing development is required by in vivo animal models of disease in order to ensure their therapeutic effectiveness as well as absence of toxicity, even after treatment for prolonged period of times. So in this case, you need proof of targeting should be there, and pharmacokinetics can be established comparatively with non-targeted therapy. Further, you also have to ensure that drug has pharmacological activity, because there are chances when you are encapsulating the drug into any nanocarrier, there are chances drug could lose its therapeutic activity. So you will have to evaluate the drug for pharmacological activity, as well as you will have to evaluate the kinetics of drug release. To understand the structural requirements for the targeting is important for its design, because to target different sites you will need different type of targeting strategies. Then you also need to sometimes transdermal to facilitate the transdermal administration, as in the case of transdermal drug delivery system. And the last thing, that integration of drug targeting technology in the earliest phase of drug development, as many drugs active at low concentration are withdrawn from R&D pipeline due to toxicity in the non-target cell. So in this case, if you are integrating these such drugs with the targeting technology, that could lead to some of the promising applications. Next please. So students, finally we could summarize that at present, although many of the problems encountered during the development of drug targeting strategies for clinical applications have been identified, analyzed and solved. However, the drug targeting strategies should be subjected to continuous evaluation with advances in the understanding of numerous processes occurring in response to administration of drug carrier or the drugs. And further, new strategies under investigation will need to be optimized and extensively be evaluated.